Right, it looks like the numbers are beginning to tail off, as often happens with these things. So in terms of the new entrants, in which case we will kick off. So thank you so much, everyone, for joining us. This is our first exciting seminar. You'll have to forgive the exciting nature of the puns throughout. That is the name of our new instrument, our new company from Exciting Instruments, which are running this seminar series. So the point was really to have a focus online for single molecule fluorescence methods and the amazing insights that they can yield in a number of different fields. And so over the course of the next, few however long really, we are going to be running these online seminars every 2 months. At this time, wherever that is where you are, this is the time it's going to be. And we are extremely excited and honored to welcome Professor Shimon Weiss to give this first seminar. I guess I am really a scientific grandson of Shimon, having been trained in single molecule methods largely in Achilles Kapanidis' lab, who was himself a direct product of Shimon's lab. And as many of you in the audience will be very aware, Shimon invented or conducted the very first single pair FRET experiment with Tugchip in his lab. Wasn't actually using confocal fluorescence, of course. It was a very different technique, and he may talk a bit about some of those experiments today. But Shimon has had a long and illustrious career in quantum dots and in single molecule FRET. Recently winning the single molecule prize from the Biophysical Society, and also now the Sackler prize from Israel as well. So very long and illustrious career, and for me has been a real shining light, if you excuse the pun again, of a beacon of real invention and what's possible when you kind of put your mind to things. So it's my absolute pleasure to pass over now to Shimon for this talk entitled Single Pair or Single Molecule FRET, a story of tools making. Thanks, Shimon.
Thank you, team. Can you hear me?
Absolutely.
Okay. Let me share my screen. Everybody can see my screen?
Yeah.
Terrific. Well, thank you, team, and thank you, Chris, for inviting me for this very first lecture. And before I start, I really want to talk and give credit for everybody who worked in my lab over the years. I'm going to give the story of how we came about single molecule FRET or single pair FRET, and a few examples for their utilization. First, I want to give credit for members of the lab, current and former. In particular, I want, of course, to note the contribution of Tugchip, who was really the first student to work on this topic and have by himself made a fantastic career. And Dr. Xavier Michelet, who has been with me for over 23 years, contributing continuously over many, many years to single molecule FRET. In blue, I highlighted people who had directly contribution to method development, Maxime Dahan, Ted Lawrence, of course, Achilles, Eyal Nir, Eitan Lerner, Antonio, Namki Lee, but many here on the list have done single molecule FRET experiments. So I want to thank everybody really who contributed to this story. I also have a lab in Israel, in Bar Ilan University, and these are current and former students. However, they do not work on single molecule FRET. They're working on electrophysiology and voltage sensing. I also want to acknowledge fantastic collaborations over the years with many, many people all over the world. And in particular, I want to note the contribution of Paul Selvin for the early days of the development of single molecule FRET. So my lab over the years have contributed to single molecule FRET and to introduction of quantum dots to biology. I'll touch upon work we've done on transcription, scrunching, super resolution imaging with Jörg Enderlein, a method called SOPHY, development of detectors, both with Polymy Group and with Eduardo Charbon at EPFL. This is really the work of Xavier Michelet in my lab. And more recently, we have been developing interesting pointy voltage sensors for super resolution electrophysiology. What I'm going to discuss in this talk is just the story of single molecule FRET. And I want to give you really, I'm sure that during this seminar series, you'll hear a lot about the technology and new developments and how to do things. I really want to give you a story, my own personal story, how we got about this and what was the evolution in terms of building the concept. So I did my PhD at the Technion. This is the Israeli MIT, if you want, a technical university in EE in electrical engineering, nonlinear optics. The topic of my PhD thesis was photorefractives. And toward the end of my PhD, my advisor, Dr. Baruch Fisher, established a new collaboration with a professor who just immigrated to Israel from Cornell. This is Aaron Lewis. And Aaron was working on, well, as I'll show you in a second, he was working on near-field microscopy, but he was also working on bacterial rhodopsin. And my group was working on nonlinear effects in all kinds of materials. And my PhD advisor was interested in trying bacterial rhodopsin for nonlinear optics. So this is how we got to know Aaron. And Aaron was invited by Fisher to come and give a seminar at the EE department at the Technion. That was around 1987. So here I want to touch on the work of Aaron in the early 80s. He actually started to work on this in 82, 83. The first paper comes in 84. And this is mostly with Isaacson. This was the first student working on this. And here you can see the title of this paper, Development of 500 Angstrom Spatial Resolution Light Microscope. And really, this is coming due to the invention of electron beam lithography. The E-beam lithography was started by Marian Isaacson, and here you see the first holes punched in a metal film by electron beam lithography, and that immediately brought the idea that if you can punch a really small hole in metal, you can actually use it for microscopy. So that's basically how the idea of near-field microscope started. And here you see some references. And later on, this is already 86. This is a real microscope where they actually do some scanning with this aperture. And look who is the second author on this paper. So this is really how Eric actually got involved in super resolution very early on in his career. And what they've done, they took glass pipettes and they heated those pipettes and pulled them to make this very cone-shaped needle made out of glass, coat it with metals, and that was the first really aperture for a near-field microscope. And here you can see some scans, line scans, where you see very sharp edges, which basically break the diffraction limit of light. In November 1989, I finished my PhD and I moved to AT&T Bell Laboratories. At that time, Bell Labs was really the mecca for solid-state physics. All the important developments were happening there. There were two campuses, one in Holmdel, one in Maryhill. I moved to Holmdel to work with the late Dr. Daniel Shemla. Shemla and David Miller were working at that time on quantum wells. Molecular beam epitaxy was just invented and people started to make very precise layers of gallium, aluminum arsenide, gallium arsenide, and basically start to implement very classical quantum mechanical problems like particle in a box. that was really mind-boggling, and I had to get to that group to do this type of experiments because I was really excited by this concept. I arrived to Bell Labs and I started to work on quantum wells, and during numerous discussions with Daniel Shemla, I tell him about this seminar I heard at the Technion just a few months before I arrived on near-field microscopy. He was not at that time aware of near-field microscopy. And the idea was that if we can see single excitons, we can understand exciton physics much better than doing classical ensemble level spectroscopy and microscopies. So sometimes in 1990, as we discussed more and more, we decided it's time maybe to visit Eric Betzig. Eric Betzig finished his PhD at Cornell, and he moved to Bell Labs as a postdoc, and very quickly is promoted to a member of technical staff, MTS, what it's called. So we decide to go to the other campus, which is in Maryhill, and visit him and talk to him about how to implement near-field microscopy for studying excitons. And basically what Eric tells us at this point is that the technology is not ready yet. So he really took the air out of our sail. And but later on, he actually, with Harold Hess, published exactly the experiment that we wanted to do, which was to look at excitons at low temperature using the near-field microscope. This is coming much later. He actually did have a lot of development to do to make this a practical instrument. Eventually, it was commercialized. There were several companies making this type of microscope, including Aaron Lewis, who developed a commercial system. In December 1990, my boss is moving to Lawrence Berkeley National Laboratory. This is a national laboratory on top of Berkeley campus, and offers me to join him as a staff scientist in the lab. So we moved. Basically, I was only 1 year in Bell Labs. And meanwhile, during this time, Eric Betzig in Bell Labs continued to improve and develop the near-field, or ENSOM as we call it, technology. He switches pipettes to optical fibers. He develops the shear force feedback in order to be able to approach this very delicate tip to the surface, very close to the surface, without breaking the tip. And as I arrived to LBL, I look around and I find that Dr. Miguel Salmeron and Frank Ogletree are actually experts in scanning tunneling microscope and atomic force microscope. And we start to discuss, because I really want to see excitons in real physical space on nanoscale. So we started a collaboration, and my very first student at Berkeley, David Botkin, is assigned to build an ultra-fast STM. In Bell Labs, we were working on quantum wells, but in particular, we were working with short pulses. We were doing ultra-fast spectroscopy on quantum wells. So the idea was to basically look at excitons both in time and space, and we wanted to create the scanning tunneling microscope with a femtosecond laser. So in fact, whoever visited my office here at UCLA might have seen the small museum I have, and this is one instrument from that time. This is the ultrafast STM that David built, still here in my office. We managed to publish 2 papers, but the concept was really flawed because we were limited by capacitive coupling of the tip to the surface. In any case, we were using photoconductive switching to excite a phenomena on the surface and then time gate the current and basically look at time response of the tunneling current. We managed to do this, but the resolution was limited by capacitive coupling. The second student that joins the lab is Takjip Ha, and he is assigned to build an ENSOM because we were familiar with the concept already since my PhD at the Technion, and we thought that ENSOM might be a better tool to look at excitons. And this is totally broken, but this is what Takjip built. You can still see 2 out of the 3 piezo tubes that were used to scan the sample. There was a shear force feedback with a fiber. That was the instrument that Takjip built to coat 8 fibers at the same time in the evaporator. And basically, we were set to go. So we have an instrument. It actually worked quite well. And in November 1993, as the instrument is basically finished and it's on the bench, Eric publishes a paper in Science, which was really a game changer. So this is a paper. He's able to look at single molecule at room temperature with this instrument that we just finished building. And this is a picture from that paper. You can see basically the fluorescent signal collected by the Nearfield microscope from a field of single molecules. And you can see that the shapes, what we call nowadays PSF, is different for each molecule. And the reason is very simple. Nowadays, we are talking about PSF engineering, and we are able to control all kinds of different field distribution and polarization. The tip gave this type of field pattern. This is looking from bottom view of the aperture, and this is from side view. When you scan such a field distribution over single molecules that have a dipole, you can get all kinds of different shapes of detected signals. Nowadays, we do this for, let's say, three-dimensional super-resolution imaging by localization microscopy. We put double helix or other shapes, and we are able to very accurately find the location in X, Y, and Z, or we can do defocusing imaging, a method that Jörg Enderlein developed to do the same in terms of measuring Z displacements. So Eric's paper is out, and we have an ENSOM just waiting. So the first thing we do is throw in molecules and see if we can see molecules. Although we wanted to study excitons, this is interesting enough, and we chose actually, based on the laser we had at the time, to see if we can see coumarin dimolecules. And that wasn't easy because coumarins are excited in the blue, so there is a lot of autofluorescence from the fiberglass, from the cover slip, and so on, and we were not sure we will be able to do this. But sure enough, although it was a molecule in the blue, we were able to detect single molecules. And here is a slide from those early days, which I'm not even sure that we published. So here you see a field of view with single molecules that we scan with a near-field microscope. Here you see a very nice molecule with a very nice PSF. Here you see a molecule that bleached halfway as we scan. Here you see a dark state. And here you see a blinking noisy molecule. So already in that very early time, we could see all these interesting photophysical phenomena that have occupied us for the past 30 years. So we were able to see single molecules as Eric did, and then we asked, what can we do that he hasn't done? And that was really the motivation. We wanted to do something interesting in addition to what he had demonstrated already. And the immediate thing that came into mind is to study molecular interaction. So the first thing we do, we threw two kinds of dyes on the surface, hoping that there will be some two different dyes by chance close to each other, and we could study some kind of molecular interactions. And at that time, we did not have internet to search for papers. At LBL, there was a big library in Building 50 where the director was sitting. And we basically went to the building, to the library, looked for some initial papers that talk about energy transfer, read them, go home, read the papers, come back, and through several weeks, you know, you had to read papers, mark references that you're interested in, and go and dig again and Xerox again and so on. So during such iterations, we basically get familiar with the concept of Fourier energy transfer. And then we discover the paper from Stryer and Hagoland for PNAS of 1967 that shows that FRET can be used as a molecular ruler. And only much, much later, only very recently, I discovered there is a paper that is actually preceding the work of Stryer and Hagoland. This is Wilczek and his group, and I'll show you that paper. So that came as a surprise because oftentimes in science, not the right people get the credit just because of circumstances and all kinds of different situations. So just for everybody to know, there is a paper 3 months before the Strine-Hagelman showing this distant dependence. So what is a famous Strine-Hagelman paper? This is basically a polyproline going from 1 to 12 monomers, basically building polymers at the length of 1 to 12 monomers, and putting donor and acceptor dyes and doing this famous experiment and getting this fantastic agreement with 1 over distance to the 6th power. But here is a paper from Milchik, which was really a big surprise. So they don't get the full curve as Strine-Hagelman, but they get the tail, as you can see here, and you can read this. Let me just minimize this part here. So quenching is attributed to radiation-less energy transfer between the tryptophan ionized tyrosine residues. Basically, they look at the same phenomena and publish 3 months before Strine-Hagelman. So we come to a realization that double-strand DNA will be the easiest ruler to implement first. We were not comfortable with biological molecule at all at that time. Polyproline peptides looked to us very, very difficult. DNA technology was just becoming a mainhold, and we thought that that would be the easiest to implement first. So we looked for help, how we could put 2 dyes, donor and acceptor, on about double-strand DNA. And luckily, Paul Selving just finished his PhD. He was hired as a postdoc at the Calvin Lab at UC Berkeley campus. And he already had done some interesting work with lanthanides on DNA. So he was familiar with conjugating dyes to DNA. So we went to talk to him, and basically Takchip, Paul, myself, and also another postdoc, Thilo Lacoste, Thilo Enderle, joined us in those discussions. And we started to run the idea that we will use a Nearfield microscope to look at single molecule FRET. And if we can do this, we could actually look at distant changes and therefore at dynamic structural changes. So that was the start. And we understood that this is going to be very, very important. So we had the motivation to demonstrate that this can be done. And basically that, by serendipity, I decided that I have to become a biophysicist. I was trained as an electrical engineer, applied physics, solid state physics, but the stakes were too high. It was clear that if we can do this, this is going to be very important. And therefore I had to educate myself in basic biology and chemistry. So you are all familiar with what FRET is. I'm not going to give an explanation for FRET. It's a radiationless dipole-dipole interaction, very much like on microscopic systems like radio transmitter and receiver, except that it happens on molecules. And I used to show many, many years ago a very nice simulator written by my colleague for many years, Xavier Michalet, and I hope I can do this again here. Let me see if I can do this. I think I'll have to unshare, and let me just. Can everybody see this?
Yeah, that's great. Yeah.
Okay.
Thanks.
So this piece of software simulator is maybe 24 years old. It was written in the purpose when we gave talks at the very early days of single molecule FRET, just to basically explain why this is going to be important. And I find this piece of simulation still very useful today. So let me show you what that simulation can do. If I can get it to work. That's a no, something is wrong. Just a minute. No, I'm sorry. This is not seen. One more trial. I tested this before, but for some reason it doesn't want to do it now. Okay, now I have it working. Just a minute. Can everybody see this?
Perfect.
Great. So what you see here is a two-dimensional test tube with donor acceptor tethered and fluctuating. And you see the donor acceptor signal, obviously, and the FRET gets one value, that's the ensemble. As we go to smaller number of pairs, we start to see some fluctuations, and we get FRET distribution. And obviously, when we go to one pair, we are at the single molecule FRET limit. So that was basically a tool to explain why this could be important. And I find this tool, Xavier had to entertain the migration of this piece of software through many, many iterations of software updates. But I find this still today very, very useful. All right, let me go back to the PowerPoint. Okay, so what Paul helped us with was to put donor and acceptor, tetramethyl-rhodamine and Texas red. It was a good spectral overlap. And we put this on surfaces and we use a microscope. And this is the very first paper. We dubbed this a single pair rather than single molecule. The single molecule name came later on when TechChip moved to Stanford and worked with Steve Chu and demonstrated the same on the surface. We actually did the surface immobilization first, but at that time we did not have yet a camera. So they beat us on the single molecule, immobilized single molecule FRET with a W view or dual view concept using a camera. So here is the very first demonstration that when the acceptor bleaches, the donor emission goes up. This was done initially with a full spectrum. We believe that in order to prove that we indeed see what we think we see, we have to see a full spectrum. So that was, at that time, a very, very difficult experiment using the Nearfield microscope with a fiber, taking the collected light and disperse it through a grating to a spectrometer equipped with a camera. So see basically that at 3.5 minutes, basically all the acceptor emission is gone and we switch back to the donor emission. This is that 1 minute, one frame at that transition. And we obviously saw the opposite where the donor bleaches first and then we see only direct excitation of the acceptor. And here is the first scanned image. We basically use a Nearfield microscope and we scan, and you can see that we overlay the two channels and we get green and red, and you can look at bleaching and so on. Again, you see here acceptor bleaching and donor emission goes up. So that was really a proof that we are seeing single molecule FRET. And shortly after, we demonstrated that this could be done in solution. So now we depart from the Nearfield and we use the confocal microscope. It was believed initially when Eric came up with his first results in '93 that you really need to have a very, very small excitation volume because the autofluorescence background scales like the volume of your excitation, while the signal from a single molecule is fixed. So if you make the excitation smaller and smaller, you gain signal to a background. And so initially it started like this, and the idea to go to confocal was really introduced by Jay Troutman. I think that was 1995, and basically demonstrating that even though you have a larger excitation volume compared to microscope, the background still can be made small enough and you can still have a very, very nice signal. So here's a demonstration where we do this in solution and we get bursts of donor and acceptor, and we can calculate FRET for each burst, and we can measure distances for different molecules of DNA. And here you see a very nice deviation actually from 1/r to the 6th power. This is a topic that Ben Schuler and Bill Eaton has looked into very carefully and published several papers, and which actually put in question how come Story and Hageland got such a beautiful, I think 1/r to the power 5.9. There are many, many issues, and obviously we are understanding those issues much better 30 years later. But here you can see that we could work in solution and we could measure distances, but the theory at that time wasn't good enough. And we followed with quite a well-accepted paper, which was really a suggestion for the future, all kinds of different experiments that I think all of them have been implemented over the years. So the concept was, okay, we can see distance changes, and therefore we could look at molecular machines in motion. Really, the idea was to take static structural biology. At that time, there was only crystallography and NMR as tools to solve structures. Cryo-EM came only later. And if we are able to measure changes, we can look at moving parts in those machines. So let's assume you start with a known crystal structure of one state, but then you change conditions, or you're looking at fluctuations in equilibrium, and you can see those. So again, this is taken from a very old slide. Okay, this is working. The simulation from Pande group is not working, but this is again a slide I used to show many, many years ago to explain conformational changes. At that time, using folding at home, the group of Pande was able to simulate folding of a short peptide. Nowadays, of course, with modern tools, the molecular dynamic simulations are in a totally different place. This is from Oster group explaining F1, F0 ATPase conformational changes during production of ATP. And basically any enzyme that runs on DNA, translocation, and changes in conditions. So it was clear that that would be useful. And obviously, we and many others were thinking in terms of the central dogma of biology. There are two major enzymes involved in basically taking the genetic code into actual proteins that do their job. So we are talking about replication, that's the DNA polymerase, transcription, RNA polymerase, and translation by the ribosome. And you have the edit degradation. All of these enzymes and processes are extremely important, and they all have several Nobel Prizes. We chose to work with RNA polymerase, and this is because an early discussion I have with Richard Ebright from Rutgers, which really got us to think how we could help in trying to solve some open questions regarding transcription. And over the years, ever since, we have been involved in really studying the initiation of transcription. So here you see the full transcription cycle of bacteria, E. coli in this case, and has concentrated over the years just on this part. I'm not going to explain the whole cycle. Many of you are very familiar, but what I just want to say that we are focusing on the initiation, which is close complex binding to promoter, opening the bubble, abortive initiation, and early elongation. So these are the processes that we have been focusing on in my lab. However, we first had a problem to solve before we are ready to attack some interesting question in transcription. And the problem is when we do single molecule experiments in solution, we cannot distinguish between donor-only labeled molecules or large distance for donor-acceptor pair. So the usual single molecule FRET in solution, molecules diffuse through the confocal spot. We collect signals on 2 APDs, and we get those bursts. Each burst is analyzed to get a FRET value, and you can get a histogram. But the problem is that you can see very nicely short distances. You see here 1 peak. So this is a mixture of 2 DNA molecules, 7 and 14 bases apart. This is from 1999 paper. But there are always, by definition, when you hybridize those molecules, you always have some free donor only, and it's very hard to distinguish between donor only molecule or a very large distance pair of molecules. So we want to solve this issue, and the idea is to add another laser. So instead of having molecule going through the confocal spot and giving us only 1 histogram, the idea is to have very fast alternation between 2 lasers. We excite the acceptor only during the red periods, and that allows us to build a 2-dimensional histogram with a new axis that we call stoichiometry. We played with this, and the initial suggestion was a little bit different than what we ended up with, and we played a little bit with the different terms of the fluorescence during the different periods. That was done by Achilles, and we came up with the ES histogram idea. And that really allowed us now to separate different species and just concentrate on species that we are interested only in them. So we can discard the acceptor only and the donor only, just keep the FRET relevant molecules and look at changes in them. So this is just an example using the transcription machinery that we can indeed do this. So here you see, here you see the open complex, or what we call ITC2, and you see this population with low FRET. You step the enzyme forward 11 bases, and you see that this cloud is moving to the right, and then you chase, and basically the whole system dissociates, and we are losing all these events. Namki, who visited us and worked with Achilles at that time, came up with a brilliant idea of how to solve another problem, which was the famous notorious gamma factor. So the idea was to put 2 different samples in the same test tube, build this Alex histogram, and actually use fitting between those 2 clouds to find what the gamma parameter is, and that allows us to come with a formula to how to extract accurate FRET. This is only halfway to finding real distances, and here I would like to mention the fantastic work over the years of the group of Klaus Seidel, really taking these tools all the way to actual, very accurate distances. Okay, so I just want to go through one example of utilization of this method, and this is our paper in Science from 2005, the work of Achilles. The problem was introduced to us by Richard Ebright, following actually experiments done in this building that I sit here today by my colleague Jay Gralla, who then many years later retired. He did experiments in the early 1980s using conventional tools like footprinting during abortive initiation, and he got some really puzzling results at that time, which was the protection of the enzyme over the promoter DNA had this very unique pattern that was not really understood. The trailing edge was sharp and the leading edge was smeared, and he got all kinds of transcripts of different distances. And that was really not understood. And over a period of 30 years, there were several different models proposed. So we set up to do this, and the 3 models that were proposed until our experiments were inchworming, scrunching, and transit exertion. So these are the models that explain this observation by Jay Gralla of the abortive products during abortive initiation. In abortive initiation, what you do, you basically provide the enzyme, the whole complex, with a subset of nucleotides. You starve basically the enzyme such that it never gets to elongation. And one proposal was that the trailing edge of the enzyme is fixed on the DNA and the leading edge is moving forward like a spring and then snaps back. The other proposal was that it's not the protein that changed structure, it's actually the DNA. So the DNA is pushed into or pulled into the enzyme. The short transit is lost and it snaps back and it repeats itself many times until by chance it moves into elongation. And the last concept was transit excursion. The enzyme sits most of the time at the back position that make very fast forward moving translocation, loses the transcript and snaps back. So no conformational changes to protein, no conformational change to DNA. It's just a question of ensemble averaging, because on average, when you look at the protection on such fast processes, you'll see a sharp edge on this end and smeared edge on the other end. So now that we have the single molecule FRET tool in our hands, we can really ask very simple questions. We can look at translocation moving forward downstream DNA. So we put a donor on the leading edge and acceptor on downstream DNA. In fact, all three models predict that this distance will change. We can then put dyes on the trailing edge and the upstream DNA. And here, basically, inch warming and scrunching predict that there is no change, but transient excursion predict that there will be change. And the last part is to put donor acceptor on DNA and test for changes in the length of the DNA. So we have done these experiments, all of this with many controls, many different positions. This is a summary of all these. And very clearly, we could show that only one of those three models is correct. That's the scrunching model. And this is really the very well accepted mechanism for abortive initiation nowadays have been confirmed many times over in many organisms. Another phenomena which was really puzzling, and we came back to it many, many years later, is that something happens at position 7. So basically, this is when the transcript is long enough to just hit the entrance of the exit channel. And we hypothesized then that there might be stress building up, and the abortive mechanism really is the transcript is lost and the strain is released, and you try again and again and again. But basically, there is some kind of blockade at position 7. And only if by chance you gain enough thermal energy, somehow this to that exit channel is opened and you can move forward. So that was proposed then. Many years later, Eitan Lerner in my lab and Achilles also did a very similar experiment for immobilized transcription complexes. We were able to go back to this question. In fact, we used here single molecule FRET only as a readout of an ensemble experiment, purely ensemble experiment, where we're looking at the kinetics of basically promoter release. And we see that if we start in positions 7, the kinetics is much more slower compared to if we start at position 0, 1, or 2, if you want, where we have much faster kinetics. And if we basically use a molecule called GRIA that is known to snip off RNA that is backtracked, we basically recover this very fast kinetics. So that really was an evidence for pausing with backtracking in initiation that was not known before, which is hypothesized to participate in gene regulation, another layer of regulation, in addition to all the other layers that we are familiar with. And that allowed us to refine the model of abortive initiation and add this other block here, which talks about this backtracking with pausing in initiation. I want to conclude my talk just by talking about where we are going. First of all, this is a review after a little bit more than 2 decades of single molecule FRET experiment that we wrote in 2018. And more recently, together with many people in the community, there was a position paper that read about the challenges in the future, which I really recommend for many of you to read. The adoption of single molecule FRET is quite remarkable. So this is taken from Web of Science. And there are about 4,000 publications that report on single molecule FRET experiments. There are about 65,000 citing articles, and single molecule FRET as a term is cited more than 116,000 times. So the adoption is good, and I claim that there is still a lot to be done and very bright future for single molecule FRET. So Tim, I think, has introduced a beautiful concept of making this whole technology much more available through open source, that people can build their own instruments. And by now, there are several commercial systems. Pico has been there for quite some time. Achilles company and Tim's company are all offering now relatively affordable and simple machines that I'm really looking forward to be disseminated and many more people to join this technology. There are many new interesting concepts that have been introduced. Just for our own work, we have been working for many years on higher throughput single molecule FRET by having many confocal spots and spot array detectors, single molecule FRET in live cells, biodetection, diagnostics, drug discovery. TechChief just published a paper, but many groups have been working on more than 2 colors, including our own lab. There are quite a few papers talking about basically having 3 or even 4 colors and getting more observables and more distances. Fantastic development, again, by several different labs, all former students and postdocs of W.E. Warner, using the Able trap together with single molecule FRET, getting fantastic signal without molecule immobilization on surfaces, very long trajectories, very good resolution in histograms. So I think we'll hear much more about Able FRET in the future. Work from Ben Schuler using the zero mode waveguides in the context of FRET and FCS to get fantastic time resolution all the way from almost picosecond, I mean nanoseconds to hours. Again, this technology I think we'll hear a lot about in the future. And a little bit of self-advertisement. This is really the work of Eitan Lerner and his group and Paul Harris. My contribution to this paper is really minimal, but trying to do what is done usually on the surface. They took the concept developed in Gilad Haran lab and improved on this even further, basically doing multi-parameter photon by photon hidden Markov modeling, where now you can look at different states and rates of transition, something that is usually done by hidden Markov on trajectories acquired on surfaces when molecules are immobilized. This is now can be done in solution, and you can look at multiple states and transition between the states in solution-based work. So this is also a very interesting development. And lastly, I just want to encourage everybody to go to this website, fret.community, and register. I think that this is really very important concept where the whole community is coming together, share tools, disseminate, and basically learn how to do things better. So this is what I had to tell you today. I again want to my current and former group members, Xavier Michelet, who has been working with me for many, many years, and his contribution is priceless. Current and former collaborators, funding for many resources, Tim and Chris for this invitation, and thank you for your attention. I'm happy to answer questions.
Absolutely brilliant. Thank you, Shimon, for that historical and personal perspective of a tool that, you know, many of us are basing our careers on and making many very interesting discoveries. So we have a number of pre-submitted questions, which we'll come to in a moment. from people who are in the seminar. But if anyone would like to ask a question now of Shimon, do you want to just identify yourself in the chat, and then we'll come to people as they type. You don't need to type the question in the chat. You can just say you have a question, or in fact, you can raise your hand.
Yeah, just hand raise, open your mic. That's fine.
Yeah, hand raise and open your mic. While we're going, I've got a couple of. Let me sum up a couple of the questions that we had submitted in advance, and I think you've covered this a little bit, Shimon, in the last couple of slides of your talk. But there's quite a few people interested in, where you see the method going. What's the future, both in terms of the methods that are going to be developed here, but also which are the areas of science that are going to have the most, that FRET and single molecule methods are going to have the biggest impact on. Okay, so indeed, the last few slides were an attempt to touch on this a little bit, but I can expand a little bit. I think that the work that Klaus and others are doing in terms of extracting structures, full structures from multiple distance constraints is extremely important for systems that you cannot solve any other way. Obviously, cryo-EM, and nowadays even dynamic cryo-EM, where you can take through microfluidics multiple freezing samples and basically deduce evolution of structure. What Joachim Frank is doing, for example, I think is very important, but FRET is probably going to compete with this. In fact, in our own lab, we have a project where we use microfluidics to look at non-equilibrium reactions and extract distances as function of time, and then solve structure as function of time. I think that is important. Static structures is important. So obviously, structural biology is still going to be the main area of application, structural biology and enzymology. But we have seen a lot of work on photophysics, on polymers, light emitting polymers, light harvesting polymers. It's hard to really tell the future, but I think that the method is so powerful that it's here to stay, and it's just up to you guys and your imagination to find new applications. Brilliant. We've got a pre-written question here from Danielle Groves. Danielle, do you want to ask your own question, given that you're in the audience? Do you want to unmute and ask?
Yeah, I can do. I just wanted to ask about if you have any advice for sort of designing these experiments. I'm a PhD student, so yeah, just in terms of sort of coming up with new experiments.
I'm not sure I understand the question. You're talking about designing new experiments on how to go about a biological experiment, or some new concept?
Yeah, I just mean sort of techniques when trying to use smFRET. Like, yeah, if you have any tips for things that you can consider.
Yeah, absolutely. So the most important part is the control experiments, preparing good samples. And what that means is that 95% of your work is ensemble level spectroscopy. You have to work really hard on getting the labels exactly where you want them to be, to characterize the purified molecule, purify and characterize the molecules and know that you exactly have what you want and there's a pure population, and do lots of control experiments. First of all, ensemble FRET, and if you're, for example, interested in surface immobilized experiments, I've always argued that doing a solution-based ALEX as a control will be a very, very good control. So you can characterize your donor only, your acceptor only, your FRET relevant populations, get a lot of idea. And those are much easier experiments. And once you get to the surface, you're ready and you're not wasting time, and you're just focusing on what you know is going to work. So I think thinking hard on the experiment, getting good molecules, well-characterized molecules, and lots of control experiments.
Thank you so much.
We've got A question from Johan Lee. Are you in the audience, Johan?
Yep, I'm here. Thank you.
Brilliant, go for it.
Yeah, thank you, Shimon. I think my question is pretty similar to what Tim just covered now. I was wondering, as the cryo-EM, develops so fast, even in the structural biology regime, where do you see the single molecule FRET, or in broad sense, other single molecule techniques will actually complementary this, as you just as before? Yeah.
All right. So just for you, let me go over the project that we have in my own lab. So as you have seen, we are working on transcription initiation. So we made, together with our collaborator, Sergei Borokov, a library of single mutant sigma transcription factors. That's a housekeeping factor for E. coli, where we then build complexes with sigma labeled at different positions. We have a library of promoters with different promoters, and for each of these promoters, donor or acceptor at different sites. We have a microfluidic, single molecule microfluidic device that Ben Schuller helped us develop. And the idea is to do non-equilibrium experiments, screen the whole library for each of the promoters, and get multiple distances, but now you get the distance as function of time for a non-equilibrium reaction. You have several crystal structure along the pathway. So you have core, you have holoenzyme, you have closed complex, open complex, ITC4. But ITC7 you don't really have. And this is where things are happening. This is where you have on pathway short-lived intermediates with large conformational changes. This is very hard to capture. So the concept is that if you are able to capture this type of short-lived intermediates with single molecule FRET, you can get, for each time point along the reaction, you can get multiple distant constraints. You have a structure in the PDB of, let's say, open complex. Now you can run molecular dynamic simulation with a known structure, with all your distant constraints, and solve the structure for each time until you get to ITC4. And then again, you start at ITC4, you have distant constraints, and you solve structures function of time. So the idea is getting lots of distant constraints as function of time in a non-equilibrium reaction, and using fancy molecular dynamic simulation with all kinds of constraints to solve structure. I strongly believe that we could solve down to atomic level structure a huge 0.5 megadalton complex and look at the moving parts, which are very short-lived. I think that will be very difficult to do with any other method.
Thank you, Shimon.
It's a lot of work. Sorry?
Yeah. No, I'm just saying thank you. That's very impressive for the non-equilibrium part, especially. Cheers.
Brilliant. Are there any more questions for Shimon, either from before or in the chat? In which case, it just leaves me to thank Shimon again for an absolutely fabulous opening seminar in our seminar series. Brilliant to see the history and the personal level of science that sometimes get lost, I think, in the sort of more standard talks that a lot of us see. So it was absolutely brilliant to see that, and we're really excited that you've kicked off this whole session for us. We will have